Greetings to all. This video lecture is going to educate you about the cell cycle. Cell cycle, it's a process which takes place right from bacteria to humans, plants, etc. So coming to the definition, cells divide into two daughter cells through a series of events that takes place in steps. A cell cycle is thus a sequence of events that cells it goes through as it grows and divides to produce a new cells. Therefore, it can be also called as life cycle of a cell. So a eukaryotic cell has two important phases, which is interphase and M phase, which is also known as mitotic phase. It also has a question phase, which is a resting phase, also called as G0 phase. So coming to the interphase, it has three phases. First one is G1 phase, second is S phase, and third one is G2 phase. During the G1 phase, the cells, it increases in size. So the increase in the cell contents is due to transcription and translation process where more number of proteins are produced. And also for the further processes, there are a lot of ATPs required, so which is also produced during the G1 phase. And also the very important thing is this is the preparatory phase for the further M phase, and it is the longest phase which takes place about 23 hours. Next is the S phase of the interphase. During the S phase, DNA replication occurs. So the 23 pairs of DNA, it duplicates into another 23 pairs. So this should be carefully monitored, and it is catalyzed by various replicatory enzymes and also various CDK cyclin proteins. And after the S phase is the G2 phase, which is also a growth phase where more number of proteins are produced, where more number of checking it occurs, because M phase is a very crucial phase and it has to be completed within 1 hour. So in G2 phase, most of the checkpoints are present and most of the regulatory proteins are synthesized. So here we can see about the interphase. So here during the S phase, there is also duplication of the centrosomes. So, and also the spindle fibers, it starts to appear. So spindle fibers are nothing but they are formed from microtubule protein. So final phase is the M phase. M phase in turn is classified into four phases. The first one is prophase, second one is metaphase, third is anaphase, and fourth one is telophase. And after the M phase, there is complete cell division takes place, which is cytokinesis or karyokinesis. Okay, and so it is the nuclear division in the mitosis. The nuclear division period, it has four phases, and also after the four phases, the cytokinesis is the complete cleavage of the cells. So coming to the prophase, which is the first phase of mitosis, so here several events occur to provide access to the chromosomes in the nucleus. The nuclear envelope starts to break into small vesicles, and all the cytoplasmic organelles, they are all converted into vacuoles, and also the cytoplasmic contents, they get doubled and they are all packed into vacuoles, and the nuclear membrane, they get disintegrated, because so that the chromosomes, they get exposed to the cytoplasm, and it shouldn't be attacked by any enzyme. So completely the chromosomes, they get exposed to the cytosol, and the cytosol will be empty because all the cytoplasmic contents will be converted into vacuoles. So here we can see the complete disintegration of the nuclear membrane. And the next phase is the metaphase. So during the metaphase, the very important process, the mitotic spindle occurs, and more importantly, in the this mitotic spindle, they occupies the poles of the cell. So because the chromosomes, it should be attached to the mitotic spindle, and also the chromosomes became more condensed and visually discrete. So each sister chromatids attaches to the spindle microtubules and centromere via a protein complex called the kinetochore. So the kinetochore attachment is very, very important during this phase. And also the chromatids, the sister chromatids, they are aligned exactly at the center point, which is called as the midway equatorial plane. The sister chromatids, they get tightly attached to each other. The next phase is the anaphase. During the anaphase, the most important part is the pulling apart of the sister chromatids on either side takes place. Each chromatid, now called a chromosome, is pulled rapidly toward the centrosome to which its microtubule was attached. So the cell becomes visibly elongated and non-kinetochore microtubules slide against each other at the metaphase plate where they overlap. And the final phase is the telophase. All the events that set up the duplicated chromosome for mitosis during the first 3 phases are reversed. That is, the chromosome reach the opposite poles and begin to decondense. That is, they come back to the original position, and the mitotic spindle are broken down. So they get disappeared, and they will be converted into actin filaments, which is the cytoskeleton filament of each cell, and the nuclear envelope it forms around, and all the vacuoles they combine to form the respective organelles. And coming to the next, so this is the end of the mitotic phase. So the final phase is the cytokinesis. So in cells such as animal cells that lack cell wall, cytokinesis begins following the onset of anaphase. A contractile ring composed of actin filaments just inside the plasma membrane at the former metaphase plate. These actin filaments pull the equator of the cell inward, forming a fissure. So this fissure is also known as crack. This is called as cleavage furrow. This furrow further condenses, deepens, and completely the cells gets broken down into 2 parts. So you can see here, so this is the cleavage furrow. The cells are broken down into 2 parts. So with this, the cell cycle comes to an end, and the cell will be ready for the next phase. So thank you.